Welcome to another episode of Tom Kennedy Science. I'm, of course, your host, Dr. Tom Kennedy. Now, today is part 5 of my series on photosynthesis. What we're going to talk about is how plants are adapted to dry environments. So, specifically, we're going to talk about C4 and CAM pathways. Wait, C4 and CAM pathways, what exactly does that mean? Okay, let's take a step back. You know me, I'm always trying to take a step back here. Now, the step back is think about photosynthesis. What are we really doing here? We're going to take some carbon dioxide, we're going to take some water and the energy in sunlight, and we're going to make glucose. So we're going to transform the kinetic energy in sunlight into potential energy into glucose, and of course, release some oxygen along the way. And in my previous lectures, I've talked about how this happens. Now, there's this enzyme, and this enzyme is called Rubisco. And Rubisco is short for ribulose bisphosphate carboxylase oxygenase. So that tells you something about what this enzyme does. It's an ancient enzyme, but what it does is it takes carbon dioxide and attaches it to a 5-carbon sugar called ribulose bisphosphate. Remember, it's got 5 carbons there. That's the ribose. Bisphosphate tells you it's got 2 phosphate groups attached to it, so it's an activated intermediate. And when Rubisco sticks carbon dioxide on there, it adds it on as a carboxyl group and forms very shortly a 6-carbon compound that doesn't last very long. It immediately splits into 3 carbon compounds. And if you remember, that carbon compound, of course, is 3-phosphoglycerate, also known as 3-PGA. All right. So that's why we call that a C3 pathway, because it forms a 3-carbon compound, 3-phosphoglycerate. Okay, so if I say I've got a C4 pathway, that means my initial carbon fixation takes place and I form a 4-carbon compound. And of course, don't forget the interesting things about Rubisco. It is probably the most abundant enzyme on the entire planet, right? I mean, every single carbon atom in your body was fixed into an organic molecule by Rubisco. That's amazing, isn't it? So it might be one of the most important enzymes in the entire world. Also, photosynthesis evolved long before there was oxygen in the atmosphere. In fact, we know that oxygen is a result of photosynthesis. So when cyanobacteria first evolved photosynthesis, they did it in an anoxic environment. Rubisco evolved without oxygen. So, what does that mean? That means that if you get too much oxygen around Rubisco and not enough carbon dioxide, then Rubisco can fix oxygen to RUBP. And you don't get 2,3-carbon compounds, you get what is called photorespiration. And we know that plants have adapted to live in dry environments. And so there's problems with this, right? I mean, you get water stress. Plants rely on getting water from their roots, and they move the water up through the roots and into the leaves, and that's the source of water for photosynthesis. And then water leaves through these tiny little openings called the stomata. Okay, now the stomata, you can see in this picture, they're surrounded by 2 cells. They're guard cells that can open and close. And I'll talk about that this summer in plant and animal form and function. It's kind of cool. But the roots actually communicate to the guard cells that they're becoming water stressed and cause the guard cells to close. Now, that's important, right? Because here's what happens. The guard cells, water is constantly flowing out of the guard cells, and that allows water to be pulled up from the roots, and you can have photosynthesis. But if you're living in a hot and dry environment, you have to close your stomata to prevent water loss. Well, that does something else as well. Remember, photosynthesis is producing oxygen, right? And that just diffuses out of the chloroplast, out of the cell, and actually diffuses right out of the leaf. And photosynthesis, it also needs carbon dioxide that it gets from the atmosphere. So it's kind of cool that trees, they don't really grow from the ground, right? They actually grow by acquiring carbon dioxide out of the atmosphere. That to me is just so amazing. But what happens is if you're water stressed and you close your stomata, okay, you're conserving water, but carbon dioxide levels go down, right? And then oxygen levels go up. In photorespiration, you stick oxygen to RUBP. You do not get two 3-carbon compounds. You don't get two of those 3-phosphoglycerates. What you get instead is one 3-phosphoglycerate and one 2-phosphoglycerate. Now, I'm not trying to be confusing everybody with all these 3-phosphoglycerate and 2-phosphoglycolate. What that's telling you, that 3-phosphoglycerate is telling you that the phosphate group is on carbon number 3, okay? Now, during photorespiration, the 3-phosphoglycerate can be used in the Calvin cycle, okay? So it can continue on. But instead of having two of them, you only have one because 2-phosphoglycolate is a 2-carbon compound. It has a phosphate group on carbon number 2. And when it's processed, this releases CO2. So instead of fixing carbon dioxide into organic molecules, this is actually going to release carbon dioxide. Oh, and unlike respiration, when you start breaking down organic molecules, you don't get ATP from this. The process actually uses ATP. So you can see that photorespiration is this evolutionary relic. Not only is it costing you carbon that you fixed, it's also costing you ATP. It's costing you energy as well. So that's a problem for these plants living in dry environments, right? So even though photorespiration is this evolutionary relic, we think that it might be important for nitrogen assimilation. It might help protect the plants from excess free radicals, from lots of oxygen and a large NADPH pool. So we think that even though photorespiration is costing the plant energy and costing it, carbon that's been fixed, it might have benefits for it. However, there are other plants that have evolved, especially in the last few tens of millions of years, to deal with these dry environments. And one way they do this is by concentrating CO2, right? If you concentrate CO2, carbon dioxide, then Rubisco doesn't go into photorespiration. So the question now is, how do plants concentrate carbon dioxide to prevent photorespiration? And this is where we have our C4 and our CAM pathways. And let's start with a C4 pathway. And like I said, C4 pathway is what it's telling you is that you're going to have initial carbon fixation. It's going to form a 4-carbon compound. Okay, so C4 pathways. What it's going to do, it's going to separate the initial carbon fixation in space. So we're going to go to a type of cell. It's called a mesophyll cell. Okay, and this is where we're going to have initial carbon fixation. And then we're going to do the Calvin cycle in another cell called the bundle sheath cells. Okay, now this is important because not only do we separate this in space, we're also using a new enzyme. And that new enzyme is called PEP carboxylase. The name tells you something. PEP stands for phosphoenol pyruvate. Pyruvate? Haven't we seen pyruvate before? Cellular respiration, right? Pyruvate is a breakdown product of glucose at the end of glycolysis. Once again, here's life reusing the same molecules. It's amazing. But a phosphoenol pyruvate, what that's telling you is I've got a pyruvate molecule with phosphate attached to it. Activated intermediate, right? And then if I've got, so that's phosphoenol pyruvate, PEP, carboxylase. Remember our carboxyl group, ACE, that tells me it's an enzyme. So we're going to stick carbon dioxide onto this 3-carbon compound, phosphoenol pyruvate, and guess what we're going to form? A 4-carbon compound. And this 4-carbon compound is called oxalacetate. Oh, wait, oxalacetate? Oh, no, we've actually seen that before, too. We've seen it in the Krebs cycle. And then that's going to go into form malate. So then oxalacetate inside the mesophyll cell will then get further reduced by an NADPH. Now, here's where the concentrating mechanism occurs. Through the plasmodesmata. Well, you remember learning that, right? Plasmodesmata are connections between different types of cells. By moving the malate from the mesophyll cell into the bundle sheath cell, okay, what's going to happen in the bundle sheath cells is a carboxyl group is going to pop off and it's going to form carbon dioxide. So what I'm doing is I'm putting carbon dioxide into the bundle sheath cell. So I'm concentrating it there. And then now, once you've got that carbon dioxide inside these bundle sheath cells, guess what? Rubisco will then fix it by adding it to RUBP and forming our 3-carbon compound, right? And because we're concentrating CO2 there, guess what? Yes, we don't do photorespiration. And then when carbon dioxide pops off malate, guess what we have? Pyruvate. Then pyruvate will then get moved through the plasmodesmata back into the mesophyll cells. And guess what? It uses ATP to phosphorylate it back to phosphoenolpyruvate. Okay, so that's a C4 pathway, right? We are separating our initial carbon fixation in space from carbon fixation by Rubisco in the Calvin cycle, okay? So let me rehash this one more time. PEP carboxylase will attach CO2 to phosphoenolpyruvate and form a 4-carbon compound called oxaloacetate, which then gets reduced to malate. Okay, this then gets moved into the bundle sheath cells. Once it's there, it will lose a carbon dioxide. One of those carboxyl groups pops off as carbon dioxide. We concentrate it in the bundle sheath cells. We prevent carbon dioxide levels from getting too low there. And then we form the pyruvate. The pyruvate gets moved back into the mesophyll cells where it's once again activated by adding an ATP. So this should tell you a couple of things. The reason why C4 plants have not taken over the world and completely outcompete a C3 pathway is because with every adaptation, there's a cost. Here's the cost of a C4 pathway. You can live in a dry environment. You can do it. You can close your stomata. You can draw down carbon dioxide levels, okay, and increase oxygen levels because your PEP carboxylase has zero affinity for oxygen, has zero affinity, doesn't go into photorespiration. But once you add on a carbon dioxide to phosphoenolpyruvate, guess what you need? You have to reduce it using NADPH. That's a lot of energy. And then once you've got the pyruvate, it requires an additional ATP to recharge pyruvate back to phosphoenolpyruvate. So even though PEP carboxylase has zero affinity for oxygen, it requires extra energy. And that extra energy, if you take a C4 plant and a C3 plant and give them plenty of water, it's likely that the C3 plant could outcompete the C4 pathway. But you get in a dry environment, it can continue to do photosynthesis when a C3 plant cannot. So here's the end result of a molecule of glucose. If I'm using a C3 pathway, I only use 18 ATPs. But in a C4 pathway, I have to take 30 ATPs. And not only that, every time I fix a carbon dioxide, I'm using an NADPH. So that's going to take an additional 6 NADPHs as well. So as you can see, there's an evolutionary trade-off. C4 plants can live in a drier environment, cost you some energy. Now, there's one other type of plant that we want to talk about. And this is a CAM plant. It stands for, let's see if I can say this right, crassulacean acid metabolism. Think cactus and pineapple and other types of succulents. What these plants do is they separate their initial carbon fixation in time. So early in the morning, humidity is higher, right? So when the humidity is higher in the morning, these plants will open up their stomata and they'll take in carbon dioxide and they'll convert it to a type of organic acid, like a malate or something like that. And then the minute it gets too warm and dry, they close their stomata. And then throughout the day, what they do is they slowly release carbon dioxide that can be used in the Calvin cycle. And the trade-off here is these plants can live in really dry environments, but they grow slowly, right? I mean, these plants live in think cactus, like in Arizona and throughout the Sonora Desert or in the Chihuahua Desert. They don't grow fast. In fact, my friend took a picture of this echinocactus in Texas. It's the biggest one he's ever seen. It's hundreds of years old, okay? But the trade-off is it can live in this very extreme hot and dry environment, or even a C4 and a C3 plant has no ability to survive. So there are the evolutionary trade-offs. I hope you enjoyed this lecture on C4 and CAM pathways. And that concludes my lectures for this month, and this concludes my lectures for this semester on photosynthesis.